Hello students, today's lecture is on methods of pasteurization. This topic is included in the chapter of preservation of milk by pasteurization. Milk is almost sterile when secreted from healthy udder. The presence of natural inhibitors in milk like lactoferrin and lactoperoxidase prevent a significant increase in bacterial numbers for the first 3 to 4 hours after milking at ambient temperatures. Cooling to 4 degrees Celsius within this period maintains the original quality of the milk, and this is the method of choice for ensuring good quality milk for processing and its consumption. Pasteurization involves heating of the milk at a very high temperature for a specific period of time, followed by rapid cooling. This method increases the shelf life of the milk by inactivating the pathogenic microorganisms as well as by inactivating the milk enzymes which are responsible for its spoilage. Pasteurized milk stored the milk below 5 degrees Celsius, which remains palatable up to 10 to 15 days. Milk is an excellent culture medium as it contains all the essential elements that are required for microbial growth, and when the milk is stored at ambient temperature, the bacteria and the pathogens can proliferate in the milk. Diseases which are prevented by the process of pasteurization includes tuberculosis, brucellosis, diphtheria, scarlet fever, and Q fever. Pasteurization process also kills the harmful bacteria which includes Salmonella, Listeria, Yersinia, Campylobacter, Staphylococcus aureus, and Escherichia coli. According to International Dairy Federation, pasteurization can be defined as it is a process which is applied to a product with the object of minimizing possible health hazards arising from pathogenic microorganisms which are associated with the milk by heat treatment, which is consistent with minimal chemical, physical, and sensory changes in the product. The process is named for the scientist Pasteur, who found that the heating of wine to 50 or 60 degrees Celsius for a short time inactivated spoilage causing microorganisms without seriously affecting the quality of the wine. Pasteurization is a process of heating every particle of a milk to such a temperature for such a period of time which is required for killing of any pathogen which may be present in the milk, and at the same time there should be minimal changes in the composition of the milk, minimal changes in flavor and nutritive value of the milk, and at the same time to increase the keeping quality of the milk so that sufficient quantities of whole milk may be supplied to large population. Unlike sterilization, pasteurization is not intended to kill all microorganisms in the food because the temperature which is used in process of pasteurization is less than the temperature used for sterilization. Instead, the pasteurization aims to reduce the number of viable pathogens so they are unlikely to cause disease. So the aim of the pasteurization is to kill all viable pathogenic organisms which are present in the milk so that after consumption of the milk there should not be occurrence of any disease to the consumer. Commercial scale sterilization of food is not common because it adversely affects the taste and quality of the product. So next is methods of pasteurization. So the first method of pasteurization is low temperature long time pasteurization, that is LT pasteurization, or it is also called as a low temperature holding pasteurization, that is LTH method of pasteurization. In this method, milk is heated to 145 degree Fahrenheit, that is 62.8 degrees Celsius for 30 minutes, and after heating there should be immediately cooling of the milk at a temperature of less than 5 degrees Celsius. So in this method, milk is heated in commercial pasteurizers, which includes large closed vats or tanks, and the heating of the tanks either with the help of steam coils or hot water jackets or by continuous spraying of the hot water on the tank. Types of LT pasteurizer. The first type is water jacketed vat, which is a double wall vat in which hot water or steam under partial vacuum circulates for heating and cold water for cooling of the milk. The outer wall of the vat is usually insulated to reduce the loss of the heat. The heat exchange takes place through the wall of the inner lining. The difference between temperature of the hot water and the milk is kept to a minimum. During heating, the milk is agitated slowly by revolving paddles or propellers. Second type of pasteurizer is water spray type, means spraying of hot water on the tank. A film of water is sprayed from a perforated pipe over the surface of the tank, holding the product which is continuously agitated. A rapidly moving continuous film of water provides rapid heat transfer. Third type is coil vat type. In this, the heating or cooling medium is pumped through a coil placed in either a horizontal or vertical position while the coil is turned through the product. The turning coil agitates the products, but additional agitation may be required. The second method of pasteurization is high temperature short time pasteurization, that is HT-ST pasteurization, which includes heating of every particle of milk to a temperature of 161 degree Fahrenheit, that is 71.7 degrees Celsius for 15 seconds, and after heating there should be immediate cooling of the milk at 4 degrees Celsius. The complete process of preheating, heating, holding, pre-cooling and chilling is completed in a plate-type heat exchanger with interconnected sections to make the process continuous. In general, the conventional HT-ST system may reduce near about 90 to 99% of number of microorganisms present in milk. So pasteurization temperature versus time. So in VAT pasteurization or batch pasteurization or LTLT or LTH pasteurization, the temperature which is used is 63 degrees Celsius for 30 minutes. So the exposure time is for 30 minutes. In HT-ST pasteurization, if the temperature of heating is used 72 degrees Celsius, then the exposure time will be of 15 seconds. But if the temperature is used 89 degrees Celsius, then the exposure time is reduced from 15 seconds to 1 second. 90 degrees Celsius temperature further reduces the exposure time to 0.5 seconds. When the temperature used is 94 degrees Celsius, then the exposure time will be 0.1 second. 96 degrees Celsius temperature reduces the exposure to 0.05 second, while only 0.01 second of exposure when the temperature which is used during the heating is 100 degrees Celsius. So as there is an increase in the temperature of the heating during pasteurization, there will be reduction or decrease in the time of the exposure. Next method of pasteurization is ultra-high temperature pasteurization or UHT pasteurization. In this method, milk is heated at 135 degree to 150 degrees Celsius for 1 to 6 seconds. During pasteurization, the product passes through heating and cooling stages. After pasteurization, milk can be packed in sterile, hermetically sealed containers, and this milk may be stored without refrigeration for 2 to 6 months without affecting the nutritive value of milk. So this UHT treatment can be carried out either by indirect heating and cooling by using heat exchangers, or direct heating by injection of the steam in the milk, or infusion of milk with steam and cooling by expansion under vacuum. Drawback of this method of pasteurization is that the severe heating affects the nutritive value, because in this method the temperature which is used is more than 100 degrees Celsius, and which can affect the nutritive value of the milk. Next is effect of pasteurization on nutrients and flavor. Pasteurization can affect the nutrient composition and flavor of the food, which depends upon which type of method that is used for the pasteurization. For example, in the case of milk, if the high temperature short time treatment is used, which cause less damage to the nutrient composition and sensory characteristics of the food than if the low temperature long time treatment is used. Because in this method, the temperature which is used is less than the T.Y.B.Sc method, but the time of the exposure in case of T.Y.B.Sc method is less than the time of the exposure which is used in T.Y.L.T method. Importance of pasteurization. So pasteurization renders the milk safe for human consumption by destroying all the pathogenic microorganisms which are present in the milk. And it also helps to improve the keeping quality of milk by killing almost 88 to 99% of spoilage causing microorganisms. So next is references for the pasteurization. Here to refer a book of Fundamental Principles of Bacteriology by Saleh, Outlines of Dairy Technology by Sukumar Day, and A Comprehensive Dairy Microbiology by J.S. Yadav. Thank you.